And then towards the end of the talk, I'll talk more about work that we've started to do more recently, and also where we see all this going in the near future. So we started this, I guess, adventure in a way, thinking about this very, very broad question about how the state of the body affects our perception of the world around us. And I know this is a sort of a very broadly phrased question, but hopefully, it'll get a little bit more specific as we go along. So to illustrate what I mean here, I'm going to give you this cartoon example. In our environment, we have these visual objects as these ones, and then through learning and experience, we may learn to associate these visual objects with very specific outcomes, as in this sort of iconic case. And if we see these food predicting cues, these learned food cues, when we're hungry, they may elicit this type of behavioral and autonomic response. Whereas if we see the same cues when we're sated, we know what they mean. So cue outcome associations are stored in our brain. I mean, they're not erased by satiety, but for whatever reason, they don't elicit the same type of response. And I mean, this phenomenon is very trivial, sort of at the behavioral level. It's obvious, and we've known about it for a long time. But what we were struck by is how little we know about the actual neurobiological mechanisms, how this is actually implemented by our nervous system. How does this actually happen, basically? So again, this is still very broad. I'm going to slightly narrow it down using 3 quotes that I really like, sorted chronologically. So we'll start with the oldest one, attributed to Apicius, the Roman gourmet, about how the first taste is always with the eyes. And this really, I think this is probably the most relevant one for this talk. This really speaks to how our distal and proximal senses interact in a way that even information from our distal senses can sort of make predictions about events in our proximity, in our proximal senses. A more recent quote from Don Quixote is, "Hunger is the best spice in the world," which again can be phrased in sort of scientific terms as caloric deficit affects our gustatory perception, if you will. And then the most recent quote is, "Hunger makes a thief of any man," which really speaks to the motivational aspects of these deficiency states, how caloric deficiency can induce a very specific motivational state. And I mean, these refer to hunger and to caloric deficiency that induces hunger, but I want to emphasize the fact that this is common to all, as far as we know, physiological deficiencies. For example, fluid deficiency will have similar phenomena, will cause similar psychological and potentially neurobiological phenomena, and other need states, for example, if you're addicted to drugs and withdrawal might have similar effects, and I'll be happy to unpack that more at the end. So we think this is probably sort of a very general phenomenon that underlies a lot of our behavior in different need states, quote unquote. So we wanted to investigate this, and I mean, you can consider different approaches. You can do sort of a broad survey of brain-wide activity. There are different ways to start getting at the neurobiological mechanisms, and we considered a lot of these. We did a lot of reading and thinking, and during all this thinking, we were sort of struck by this part of the brain, the insular cortex. This was not the original plan, but surveying the literature, we were sort of struck by this part of the brain that has this, I think, of this intriguing characteristic of being a place where the internal meets the external. And what I mean by that is that insular cortex is an anatomical distinction. Functionally, it encompasses in the mid and posterior parts the primary visceral cortex. In the same way that we have a primary cortex for vision and for audition, we have a primary cortex for our viscera, right? And this is inner insular cortex. So this sensory information from within our body is constantly being relayed to the insular cortex via sort of canonical pathways, brainstem to thalamus, etc. On the other end of that, we know that insular cortex, based on human studies and animal models, is highly responsive to salient cues in our environment. For example, pictures of food when you're hungry, but also faces with a strong sort of emotional expression and other motivationally salient cues will also drive a strong activation of insular cortex. So we thought this is sort of an interesting characteristic of the internal and the external, and obviously we're not the first people to notice this. This has attracted a lot of very deep thinkers in the field which have used this to develop this idea that what insular cortex actually does is combine these distal cues with internal information to compute interoceptive predictions about future physiological state. For example, if I see the McDonald's arches, how will eating that food affect my bodily physiology? So that's sort of the basic idea, and it in a way is attractive because it answers this idea of why or what for would you need an interoceptive cortex, which is sort of the general answer you usually get is this: why do you need a cortex? It's to sort of make predictions about the world and update these predictions and guide behavior with these predictions. So that's a sort of a very strong theory in the field, and I'm going to center this talk around this theory. In the first part, I'll briefly discuss our work on representation of learned cues in the insular cortex. Then I'll talk a little bit about how the internal state of the body is represented by ongoing activity in insular cortex. And then I'll try to show you some of the ways we've tested to see whether one is indeed a prediction of the other. And then, as I said in the last part of the talk, I'll discuss more recent work and also where that work is leading us in the future. So we are investigating this using mice as a model system, leveraging mouse genetics with other tools that we have these days in mice. And the first thing we needed is to create these visual food predicting cues for these mice, because obviously they don't have the experiences that we've had to create these food predicting cues. So for that, we use this simple visual discrimination task. It's a fully operant task. So if the mouse does something, something happens. If it doesn't do anything, nothing happens. And every trial, the mouse sees one of three visual cues here. They're just stripes moving in one of three directions. They're completely arbitrary, completely meaningless, and perceptually pretty similar but slightly different, so the mice can discriminate between them. And if the mouse licks the food cue, it gets a food reward, in this case a highly nutritious caloric milkshake, Ensure, that is used as a food replacement or supplement. If it looks to the aversive cue, it gets quinine, which is just a bitter tasting that it usually would like to avoid. And finally, the neutral cue, nothing happens. So mice learn to do this task pretty well within a week or two. And you can see here the average licking of a mouse licking selectively to the food cue, not licking to other cues. I hope you can see my mouse. That's sort of flickering around the screen to show what I'm talking about. So the mouse is really has a very selective behavioral response. And then when the mouse is sated, whether it's sated on Ensure, on Chow, or on anything else, these responses go away. So it's obviously motivated by hunger, or by caloric deficit, if you will. And so the first question we asked, having this at hand, was whether insular cortex activity is in any way involved in this behavior. So for this, we did pharmacological inhibition, also chemogenetic inhibition. And we found quite surprisingly or strikingly for us that inhibition of insular cortex really strongly reduced the responses to food cues, the behavioral responses to food cues in this task, without affecting the incorrect responses to other cues. And what was a little bit even more surprising for us was that despite the fact that insular cortex activity was sort of necessary for doing this task, it was not necessary for other types of feeding. So home cage feeding of chow was not affected by inhibition of insular cortex. And I think even more importantly, if we inhibit insular cortex in mice that are very, that know the task really well, they're in the task apparatus, they're in front of the screen, but the screen is just blank gray and they can have as much Ensure as they want. Insular cortex inhibition does not affect Ensure consumption. So it seems to be really somehow involved in acting on these cue outcome associations beyond just feeding per se. So knowing that, we wanted to try to see what kind of activity in insular cortex might actually underlie this phenomenon. So for that, we wanted to use an imaging approach to track the activity of large populations of neurons over these slowly varying states. They vary over hours and minutes, hours and days. So we wanted to sort of track the same populations. Insular cortex is hidden behind these essential parts of the skull and jawbone. This is the back of the eye socket here, the NZ, and the jaw. So what other people have done in the past is break these bones, but obviously that's not very friendly to awake behaving mice. So we did a little trick instead. We just stuck a little reflective microprism in, and this is sort of like a a periscope that allows us to peer into the insular cortex, in an intact insular cortex in this awake behaving mouse. And using this prep, you can see here, this is a movie of insular cortex neurons flashing away. They're expressing the genetically encoded calcium indicator, GCAM6. And I hope you can appreciate that we have pretty good cellular and subcellular resolution, so we can discern individual dendrites and axons, and we've also imaged the activity of these, as I'll show later in the talk. So this is a pretty good prep. And now, if we use this prep to image activity of insular cortex in this task, this is sort of a grand total summary of what we get. So we found that about a third of the neurons in insular cortex respond to these visual food predicting cues. Half of these neurons increase their activity here in red, half of those neurons decrease their activity in blue, and if we follow the same neurons, a few hours later or even a day later when the mice are sated, their activity or their cue responses completely flatline. And this was very different from what we previously observed in other parts of the brain, for example, in visual association cortex, where the responses still persist, but they're modulated. And then we follow this up by doing a separate study in a separate group of mice where they're now doing the same task for water instead of food. So it's the exact same task, but now the mice are thirsty instead of being hungry, and the reward is a little drop of water instead of Ensure, and we basically see the exact same thing. And then we did a series of experiments to test whether this is really due to the physiological state of the body or to maybe something else. There are other important variables to consider, for example, arousal that might change or does change with the transition to hunger, from hunger to satiety, licking and preparatory facial activity changes. So we did a bunch of experiments and actually found that if we control for these variables, the effects are even larger. So we came to the conclusion that it is likely the actual physiological state of the body that's driving this big change in cue responses. So then the question is, how does this actually happen, right? How do we get from a physiological deficiency, whether it be calories or fluids, how do we get to actually modulating cortical activity? And, you know, we sort of had an appreciation of hunger as this really fascinating multifaceted phenomena that involves various secreted factors from, you know, from our fat tissue, from our gastrointestinal tract, from our liver. There are peripheral sensory neurons innervating these tissues. There's an interaction between the peripheral sensory neurons and the hormones. So there's a lot of things going on. So as a first step, we decided to focus on a pretty well-known bottleneck in the system, these so-called hunger neurons in the hypothalamus. And we have genetic access to these neurons in mice because they express the peptide, the gene, agouti-related peptide. And so we can target them specifically in mice. And using this approach, others before us, our labs and others, have shown that these neurons are sort of necessary and sufficient for feeding in the classical sense, that if you ablate them, adult mice will starve to death. If you activate them in a sated mouse, it will start eating voraciously. So this seems like a sort of useful access or entry point into the circuits that mediate hunger. And we can do the actual same thing with thirst. There are different signals for fluid deficiency for both blood osmolarity and blood volume. Also blood pressure regulates these. And again, we can focus on an important bottleneck, which is this system in the hypothalamus is 3 interconnected nuclei called the lamina terminalis. And we focused on manipulating 2 of the nuclei here, the subfornical organ and the median preoptic. And what we have here actually is sort of a way to affect in the hypothalamus both sensors and actuators of the response to this physiological deficiency. So here's an example of what we did with that. So this is the average licking of a mouse, licking selectively to the food cue, not licking to other cues. And when this mouse is sated, it doesn't lick to anything else, or it basically does nothing, sits there and basically waits. And then in the same mouse, we put chemogenetics in AGRP neurons, so DREADs, which are receptors that will only respond to a exogenous ligand. I hope you can see here these teeny tiny red triangles at the bottom as where the AGRP neurons are in the basomedial hypothalamus. So now if we activate AGRP neurons in this sated mouse, the mouse now re-engages in the task, starts licking selectively to the food cue, not licking to other cues. So it's really like we sort of flipped on this motivational switch, and now the mouse will do whatever it needs to do to get food, really whatever it learned in this context that it needs to do to get food. So it's really quite a specific behavior. And in the same mouse, we can also image the activity of insulocortex neurons across these natural and artificial hunger states. And here's sort of a grand total summary of these experiments. So we see, as before, neurons excited by the food cue, suppressed by the food cue in red and in blue. The responses of these same neurons flatline when the mice are sated. And then I'm adding here in black these curves, I hope you can see, which are the responses of the same neurons when we activate AGRP neurons. So responsiveness, as you can see, is completely restored. And we also did more sort of elaborate analyses of population activity patterns. And basically, as far as we can tell, the cue response patterns in insulocortex look as if the mouse is naturally hungry. So it's really quite a powerful way to mimic hunger, at least with respect to cue responses. That's like a little teaser for what will come up next. But for now, I'll, to keep the momentum, say that we basically were able to do the exact same thing with thirst. So here's a mouse licking to the water cue, not licking to other cues when it's thirsty, not licking to anything after thirst is quenched. And now we activate a small population, this teeny tiny nucleus here, the subfornical organ. We activate these thirst neurons. And again, the mouse re-engages in the test, starts licking selectively to the water cue, not licking to other cues. So we really activated now. We have sort of two switches that we can activate. We've activated now a different motivational switch, and the mouse will do whatever it needs to do, or whatever it learned that it can do in this context to get water. And when we image insulocortex in these mice, we see again responsiveness is completely restored to water predicting cues. So if you think about it, I started this talk with this sort of very broad idea of, how does the state of the body affect our perception of the world around us? And we've sort of gradually now narrowed it down to how does the state of the body affect cortical activity or insulocortex activity? And now we've narrowed it down even further to sort of operationalize it to be how do hypothalamic neurons affect cortical activity, right? So we've sort of now made this question operational, and now we have two neural components, and we have a whole suite of neuroscience tools that we can use these days and just connect the dots between these two neural components to answer this question. And so this is what we did. I'm briefly summarizing a series of circuit mapping studies that we did. We started with an unbiased approach of using this genetically engineered HSV, herpes simplex virus, engineered in David Anderson's lab at Caltech. And this jumps forward synapses to label a pathway from a genetically distinct population. So we use this as sort of to show us all the possibilities downstream of AGRP neurons that are relevant for the study. We then followed that up with rabies, pseudotype rabies monosynaptic tracing, followed that up with channel rhodopsin circuit mapping and slices to make sure that we're looking at real synapses. And then I think importantly, we followed that up with in vivo studies where we manipulated specific components in the pathways that we were identifying and looked at how that inhibition affected insulocortex activity and behavior. And sort of a box and arrow summary of these studies is that we found that hypothalamic hunger neurons affect insulocortex activity via this limbic pathway from this somewhat obscure midline thalamic nucleus called the paraventricular thalamus to the amygdala, and specifically the basolateral amygdala. And we were very excited to find that thirst neurons actually converge on the same pathway, even at the level of individual neurons sometimes, to affect insulocortex activity. And because we see sort of this convergence between hunger and thirst, we speculate that other hypothalamic systems that drive other motivated behaviors might actually converge on the same pathway. We speculate that this might be a common theme. For example, based on anatomy, we know that neurons that drive the temperature to seek, drive the seeking a cool temperature when you're too hot, converge on this nucleus. The same thing for parental behavior and aggression. So we think this might be a common theme. But now to take a step back from this, the way we think about it is that the amygdala acts as sort of like a saliency filter of the outside world for the insulocortex. This is based on our work and work by other groups, most notably that of Alfredo Fontanini. And we think this saliency filter, based on our work, is gated by hypothalamic neurons. So this deals with the sort of the circuitry that underlies food cue biased responses and hunger or thirst dependent gating of these need relevant cues. but it doesn't say anything about the internal, because as I said at the beginning of the talk, insular cortex is this interoceptive cortex that responds to things. For example, we know that insular cortex responds to, even individual neurons respond to gastric distension, nutrient absorption in the intestine, blood pressure changes, dryness of the mouth and esophagus, heart rate changes. All of these things affect insular cortex activity. And so this brings me to the second part of the talk, where we describe how these physiological deficiencies might be encoded in ongoing activity in insular cortex. So I have to say that what actually sort of compelled us to look at this, we were thinking about this for a while, but what actually sort of drove us to look at this more carefully was this nagging observation that there was always a lot of activity in insular cortex. Whenever I would put a mouse under the microscope, we would always see, let me play this again, this ongoing activity that was sort of the insular cortex was just very active. And this is in line with the fact that other people have reported an average spike rate of about 8 hertz at the insular cortex. And this is very high compared to other sensory cortices. And we sort of fantasize that this may have something to do with the fact that this is a sensory cortex of the body is always on. So maybe this is actually reflecting the constant flow of sensory information from the body. That was, this sort of speculative hypothesis, but as sort of a very first little baby step in the direction, we just looked at the activity when mice were sitting there and doing nothing under the microscope, thirsty or after their thirst is quenched. And this is an example of 2 neurons. You can see that the neuron on the left here has a lot of activity and then less so when the mouse is quenched, and the neuron on the right side has the opposite pattern. And so this really drove us to look more carefully at how ongoing activity might change as hydration is gradually changing. And I say hydration because thirst was a good starting point for us because it changed relatively rapidly within a couple of hours. So we could really look at things as they're changing. So to do that, we developed this approach of looking at interchild intervals. So this is the same visual discrimination task that I showed you before for cue responses. The cues are color-coded here with these colorful rectangles, and then the gray is the interchild interval where the mouse is basically waiting for the next child to start. And then we sub-selected a subset of these interchild interval when the mouse is basically doing nothing, it hasn't had a reward in a while, it's basically sitting and waiting for the next cue to come on, hoping that it might be the water predicting cue. And if we look at these, we wanted to sort of zoom out in time and not look at them at sort of second or millisecond resolution, but actually look over many minutes or hours. So this is the cumulative water consumption of a mouse in this task over an hour and a half. This is 3 half-hour sessions just back to back. And you can see the mouse is gradually accumulating water, getting more and more and more water. Every third cue on average is a water cue, so it's getting more and more water. And after about 45 minutes, the mouse's thirst is quenched, it's done with the task. And then you see after the mouse is quenched, basically this line is mostly flat, so the mouse is just basically sitting there and waiting to get back to its home cage. And if we look at activity during this time, we can find neurons like this one that has this ongoing activity. So again, this is sort of the background activity that's task independent. This is the spontaneous or background activity, or what we call ongoing activity of this neuron. And you can see that it's high initially and decreases gradually as the mouse's thirst is quenched. Here's another neuron that has the opposite pattern, has some activity that actually increases. So the top one would be sort of like a thirst neuron. The bottom would be like a satiety neuron. And at the population level, we're imaging usually about 150 neurons, and lately obviously more. And we found basically that as thirst was gradually quenched, the average activity level across the population did not actually change. So everything was stayed relatively constant. What actually was changing that some neurons increased their activity and some neurons decreased their activity. And I hope you can appreciate here, I've outlined on the top in pink these neurons that have some activity that decreases as thirst is quenched, and on the bottom the opposite, neurons that have some activity that increases as thirst is quenched. And whatever pattern we saw, and again I'm saying a pattern because the average activity does not change, whatever pattern we saw was consistent across days, and this is just one example. We can look at that by reducing the dimensionality of the data. These data have 130 dimensions in this case because it's 130 neurons. So we can just sort of using standard methods like principal component analysis, look at the 3 first present components. And you can see thirst and quench states are separable and consistently separable across days. And it's sort of like a bottom line approach that's pretty popular these days. We can use a classifier. So we train a classifier on a thirsty versus quenched on day 1, and then give the classifier data from day 2 and basically ask, say, here's all the data, let us know what you think. Is this thirsty or is this quenched? And in this third bar here, you can see that the classifier did what I think at least was incredibly well, because in all cases, it did above chance and well above chance, between 70 and 80% in most cases. So we can really, there's some consistent pattern of activity that's different thirsty versus quenched. But at this point, we can't say much more than that. So then what we did in this recent paper, a lot of it was actually sort of dedicated to figuring out whether this really had something to do with the physiological state of the body or some other co-varying factor. So we did different experiments. We did experiments to show that this reflects hydration state, for example, by injecting isotonic or hypertonic saline to push around the physiology independent of consumption. We found that this ongoing activity transitioned to a quench state several minutes before behavior changed. So it seemed to be somewhat independent of the sort of the current behavior, and I'll be happy to talk about that more at the end. We found that it does not reflect arousal or time elapsed in session, things that insular cortex has been implicated in before. We don't see this all over the brain. We've looked in a couple of other areas along the visual system, and we don't see this. So it's not this brain-wide phenomenon. There's something that's slightly more specific. And I mention that because, for example, recent work has shown that movement-related activities are very prevalent all over the brain. But we think this is something that's at least slightly more unique, and that's something we still need to figure out how specific this is, and where do we see this in the brain and this interconnected network with the insular cortex. This is something we're very interested in. Importantly, thirst is different from hunger, or patterns related with caloric deficiency and fluid deficiency are different, put it in a different way. And I think the most surprising result for us was that this ongoing activity was completely insensitive to activation of these hypothalamic neurons. So this is something I think is really not only surprising, but very important. So I'm gonna, at least for this talk, so I'm gonna focus on that now and unpack it a little bit more. So what you're seeing here is, again, the cumulative water consumption of a mouse over a couple of hours of an experiment. So the mouse is initially drinking more and more and more and more, accumulating more and more water rewards. And you can see at this point that I'm pointing with my mouse, the mouse gets quenched. And it basically sits there and does nothing for a while as we keep imaging insular cortex activity. And then when we artificially activate thirst neurons, you see the mouse re-engages in the task and starts accumulating more water. And I want to remind you, this is the same data that I showed you before. So this is from the same mice, same data. So what's going on here is that the mouse is licking selectively to the water cue, not licking to other cues. And cue responses in the insular cortex are completely restored. Okay, but despite all that going on, the ongoing activity seems completely unaffected by this. So this was quite surprising for us. So you can see here on the top in pink highlighted neurons that have some activity levels that gradually decrease. And you can see that when we induce this artificial state of thirst or motivation for water, activity still stays down and seems insensitive to that. Whereas these neurons on the bottom have some activity that increases, and it still stays high. If anything, it even increases a little bit more. So this is really, for us, was quite an exciting dissociation between the cue responses that seem to follow more sort of the motivation or the ongoing behavior, and the ongoing background activity that seems to really faithfully reflect the true physiological state of the body, and not whatever the mouse is actually doing at the moment. And importantly, we found the same thing for hunger and for activation of hunger neurons, and I'll show a little bit of that soon. So this brings us to this point where I showed you that we have a handle on learned sensory cue responses in the insular cortex, and we have a somewhat dissociable handle on interoceptive information, ongoing activity reflecting physiological state. And we figure this puts us in a pretty sort of exciting position to test this idea of interoceptive predictions of whether one really is a prediction of the other. So this brings me to the third part of the talk. And the question is, how do you actually do this? Right, because I mean, you can think, well, it's easy. You just do sort of a Pearson correlation or whatever similarity pattern, similarity metric of the patterns. But the problem is for us that we realized that population activity in insular cortex does not only reflect thirst or hunger. It reflects things like arousal, as we and others have shown. It reflects levels of pain, of water deficit and caloric deficit, like I showed. It also reflects changes in temperature and other things. This is sort of a multimodal cortex that seems to represent different types of information at the same time. But we want to somehow isolate and look only at specific patterns that have to do with water or caloric deficit. So to do that, what we did is we considered this idea of what you can call population activity space. So if we have activity, say we record from 150 neurons, so we have activity with 150 dimensions, it's this space. And it's illustrated here in 3 dimensions as if we have only 3 neurons, which is fine. And then in this space, activity can move to different places in the space. For example, it can move in this direction, as you can see with this orange arrow, when there's a painful stimulus. It can move in a different direction when there's increased water deficit, in a different direction for caloric deficit, in a different direction for arousal. And what this approach gives us is that we can also look at things simultaneously. For example, where my arrow is the point where there's an increase both in arousal and in caloric deficit, which is actually the natural way things happen in animals, right? But then we want to look only on these specific axes and look at how predictive cues move the activity along this axis from hunger to satiety. So this is what you're seeing here, and I hope this will sort of make that explanation a little bit more complete. So this sort of cloud of activity here in blue is the activity patterns associated with caloric deficit, with hunger. And we give these, just for normalization, a value of 1. And here in yellow is this cloud of population activity patterns associated with satiety. And what we do is basically draw a line between them, as you can see, this gray line, and we just look at movement along this line between them. And so this is sort of the activity in population activity space. And so what you're seeing here is the average Q evoked activity. So times 0 is when the Q comes on in the task. And -2 to 0 is the end of the intertrial interval when the mouse is basically sitting and doing nothing, waiting for the next cue to come on. And almost by definition, you can see that the ongoing activity when the mouse is hungry is close to 1, and when the mouse is sated, it's about 1. And then when a sated mouse sees the food cue and basically does nothing, as I showed you before, the activity basically stays around 0. So this is this yellow cloud of the average activity patterns related or associated with satiety. And when a hungry mouse sees the food predicting cue, starts licking, and gets the little drop of ensure award, we found that the activity actually dips down all the way to a satiety-related pattern, and then goes all the way back up to a hunger-related pattern by the end of the intertrial interval. So, and I want to just emphasize the fact that these couple of seconds when the activity looks like as if the mouse is sated, it will eventually be stable 2, 3, 4 hours or a day later, the mouse will be sated. So the activity patterns will be these stably. But what we're seeing here is the activity sort of transforms itself or the cue and consumption transform the activity transiently to a satiety-related pattern, and then it goes back to the current level of hunger. So this was for us sort of quite encouraging, and seemed to support this idea potentially of interoceptive predictions or simulations of a future satiety state. And it made us curious about this weird artificial stimulation of hypothalamic neurons and how this might then look like. So what you're seeing here is, again, artificial activation of hypothalamic hunger neurons that I showed you before, re-engages mice in the task. They look to the food cue and they get a food reward. And first of all, you can see that the ongoing activity actually still reflects satiety, even though the mouse is actively engaged in the task. This is the same thing I showed you before for thirst, only now for hunger. So even though the mouse is engaged in the task, it's compelled by this artificial activation of hypothalamic neurons, activity is still in a satiety-related pattern. And then when the mouse sees the food cue, is compelled to lick, responds to it, and gets a little ensure award, the activity goes further along this axis to negative values beyond a satiety-related pattern. And so this to us seemed like potentially we were sort of puzzled by this. Then we thought, well, maybe this is actually sort of like a prediction of the physiological consequences of overconsumption, sort of like hypersatiety. You know, your stomach is not this full, it's actually this full, and your insulin levels will be higher, your blood sugar levels will be different, right? So we think this might be related to that. It's purely speculative at this point, but we find that interpretation kind of interesting. And we found the same thing for thirst and water cues. And so to summarize what I showed you at this part, I'm going to have this very high-tech PowerPoint animation here on the left side of the screen. So when the mice are hungry, activity is sort of, and they're doing nothing, activity is in around this sort of, around this cloud of the average pattern of associated with hunger. And then when a hungry mouse sees a food predicting cue and gets a little drop of ensure, activity transiently goes all the way to satiety-related pattern and then back to the hunger state. And I want to emphasize that the food reward that the mouse is getting here is 1,000th of what it needs to be full. So it's 0.1% of what the mouse needs to actually be full. So it's really a minuscule, it's not even tickling really the satiety of the mouse. So it's really something different that's going on here. And we were really struck by the fact that it goes all the way to satiety. We were thinking it probably will just move it, nudge it a little bit in the direction, but it really goes all the way there. And then after the mouse actually reaches satiety, and a lot of the experiments, the measurements were done actually a day later after overnight consumption of chow, the activity is stably at 0. So this is the satiety-related pattern. And then when we now compel the mouse by activation of these hypothalamic neurons to keep consuming beyond what it actually needs, the activity will go further along this axis to beyond the natural satiety. And this may seem like sort of a very artificial situation, but I think it does have real world equivalents. For example, if you had sort of a nice meal, you just had your, I don't know, let's stay with the McDonald's, you had your burger and fries, and you've had, a certain amount of it, but you're actually full and it's a nice dish, especially if you're in the US, it's probably larger than what you actually need. And you sort of sit there and you're like, well, yeah, I'm feeling full, but then you have a second look and you're like, well, maybe I can have a little more of these fries. But then you think to yourself, well, but if I do that, I'll feel really stuffed, right? So what you're doing here is you're actually predicting, right, the physiological consequences of overconsumption. And you sort of speculate that the insular cortex activity might actually have a sort of a specific prediction of the specific changes in your body. For example, gastric distension, but also other things like different hormonal levels and other things you can imagine, I guess. So the last data point for this part of the talk is yet another dissociation, I guess, between the present and the future in insular cortex activity. So what you're seeing here is now we look at the cue evoked activity in 3 different parts along the sort of path to satiation. As the mouse is gradually getting sated, we look at 3 different parts when the mouse is really thirsty, sort of halfway there, and then really just before it's getting quenched. And then you can see that the ongoing activity indeed gradually goes down from along this axis towards the satiety-related pattern. However, regardless of what the starting point is, when the mouse sees the cue and gets a little drop of water, the activity will go straight to a satiety-related pattern and then back to wherever it previously was. So regardless of what the current deficiency state is, the cue will evoke sort of a transient simulation of one specific satiety-related pattern, which is the endpoint that will arrive, a couple of hours or maybe 15 minutes later. So this is what you're seeing here, and we think this is really yet another way that suggests that really there's this functional dissociation between encoding of or estimation of the current state of the body and the predicted future state of the body in the insular cortex. So as sort of a conceptual summary, we think that environmental cues predicting the availability of food and water, for example, for the cliche example, the McDonald's arches, are sort of funneled or filtered through this saliency filter in the limbic system to the insular cortex through the amygdala, and this saliency filter is gated by the hypothalamus. Now, in parallel to that, information about the state of the body is conveyed to the insular cortex via, or is conveyed to the brain sort of via known pathways through the brainstem and hypothalamus, and sort of empirically we found that the hypothalamus was not involved in regulating the insular cortex representation of physiological state, and we sort of, we suspect it's probably the canonical brainstem pathways that are doing this from the vagus, etc. And we're currently working on experiments to sort of, drive that point home properly. And then at the end of this part, I showed you that cues predicting availability of fluid water drive a transient simulation or prediction of the future satiety state in insular cortex. And we and others speculate that this might sort of explain the involvement of insular cortex in various, it's implicated in so many different behavioral situations. It's thought to mediate the interoceptive aspects of, addiction, emotional regulation, social behavior, and we think this might be the explanation to that. And again, I'll be happy to talk a little bit more about that at the end. For now, I want to use the last few minutes of this talk to tell you about what we've done recently and where we're taking it next beyond what we've been doing now. So the way we're thinking about this is as interoception as an active process. So it's not this passive process where there's some change in the body and it's propagated to the brain, and then the brain responds to that and modifies the body. So that would be sort of the passive version of the brain-body loop. Instead, we think about this as an active process where our brain is constantly making prediction using environmental cues of the world, in the world around us, making predictions about upcoming changes to the body, and then actually preparing the body for these changes. So it's sort of an active version of the brain-body loop. And of course, this is, by no means a new idea. I don't need to obviously explain this to this audience. And there's been a lot of thinking about this. In a way, you can think that it traces back to the quote I gave from Apicius, the Roman gourmet. But I think really one of the pioneers of the experimental work on this was the famous Ivan Pavlov. And this is a newspaper clip describing his work from over 100 years ago that I really like. So it talks about how our minds digest the most important part of our food. And I'll be happy to speculate with you what that actually means, because I'm not sure. But I think the more important part about here in this title is about this Russian scientist who collects the stomach's psychic juices. And I think this is really a lovely way to describe what we now call the cephalic phase of digestion, which is this phenomenon that if we're hungry and we smell food or see food or even think of food, it will cause gastric juice release in our digestive system, gastric motility, insulin secretion from our pancreas, and obviously, as you all know, actually salivation, right? And if you think about it, what's going on here is that our brains are making a prediction about the consequences of the upcoming ingestion and digestion of food, but that hasn't happened yet, right? So it's actually affecting bodily physiology in preparation for something that it's predicting will probably happen soon based on, the fact that you're seeing the food or smelling it, for example. So what we want to do is actually get into understanding sort of the neurobiology of how this actually happens. How are these predictions formed in the brain, and how are they propagated back to affect bodily physiology? So I hope based on this talk you can sort of agree with me, or at least sympathize with the notion that insular cortex would be an interesting starting point, because it seems to have these interoceptive predictions. And so definitely for the sensory component of this brain-body loop, it's very relevant. It's also very relevant for the motor aspect here, because a lot of work, starting from Penfield, but also pioneering work by Cliff Saper and others, have shown that microstimulation of the insular cortex will affect things like gastric motility, blood pressure, breathing rate. So it affects a lot of different autonomic function. So it seems that there's a sensory and a motor component, and that's why insular cortex is considered by some in the field to be involved in both the sensory and the motor components of the system. So I think obviously this is very much in line with the idea of the seminar. That's why I'm so happy to be here and excited for the discussions that are coming up. And I really like this graphic in the website, because I think it really illustrates nicely the way a lot of us are thinking in the field, not just me, but also including me. And my little small version of this is just putting insular cortex here in the center. And the way we see this is we want to understand how insular cortex interacts with these different systems, how it makes predictions about the gastrointestinal system, the cardiovascular, the respiratory system, the immune system, how it makes predictions, and then affects bodily physiology based on these predictions. And we think there's a lot to do with this, how the predictions are being formed, how are they updated when they're violated, what are the timescales that this happens. So I think it's really a fascinating field to go into, and this is what we've started to do. And the key to the way we're thinking about this, especially from the perspective of cortical coding, is stimulus control. In the same way that the external senses or the neurobiology of external senses has really benefited from proper stimulus control for understanding stimulus representations and then predictions about these, all this idea of predictive coding, we want to basically do the same thing, and we think what we need is to get tighter stimulus control for different interoceptive stimuli to be able to do this and interpret the cortical activity patterns we're recording. So in my lab, we've started to focus on the gastrointestinal system specifically. We're also working a little bit on cardiovascular and respiratory physiology, but mostly the gastrointestinal system. We want to manipulate the activity and record. It's pretty standard these days to do continuous glucose measurements in mice with telemetric probes, but we can also manipulate the system. For example, there are these sort of traditional old school methods to do this, which I think are incredibly elegant. For example, manipulating the content of the digestive system, manipulating caloric content or separately macronutrient content. And there are newer ways to do it these days, or to manipulate the system. For example, by using the new genetic access we have to specific labeled lines of information from the periphery. So thanks to pioneering work by Steve Lieberless, who was also part of the seminar, and also Zach Knight and others, we can now genetically manipulate, activate, or deactivate specific neurons conveying, for example, stomach stretch or lung stretch, blood pressure. So we can manipulate these separately and really understand or control the stimulus, and then combine that with large-scale recordings of insular cortex activity and also manipulations of insular cortex activity, and also recordings and manipulations of long-range inputs to the insular cortex. So this example is amygdala axons, which we've published some of these data. We can record the activity, and again, we can start manipulating different components in the system and understand, for example, how the internal and the external meet in the insular cortex. What are the specific inputs, bring these different and the types of information and how they're being integrated at the level of the local circuit. And again, most importantly, how this is then propagated to the body to affect bodily physiology. So I'm going to give you just a little sort of teaser example for one of the more recent experiments we've done. This is imaging insular cortex in a mouse with a gastric catheter, and we've infused different nutrients into this mouse. But for the sake of this talk and the spirit of predictions, I'll show you just one result of what happens when infusion of sugar, of glucose actually specifically, into the stomach of this mouse is either fully predictable or completely unpredictable. So this mouse is trained to predict based on external cues the infusion. And you can see that the responses across these 80 insular cortex neurons are very different if the infusion is cued or uncued, or predicted or unpredicted. So this is really not a classical, sensory response in a way. You can see these neurons only respond when it's uncued, but actually don't respond a lot when it's cued. And this is just an example of two neurons responding. One of them on the top responds when it's cued, the other one when it's uncued. So this is the type of work we want to do to understand, and as I said, expand these to also newer methods of manipulation to understand insular cortex and its role in this brain-body loop. So if you're, at least if you're even half as excited about this as I am, I invite you to contact me and join my lab at the Weizmann Institute. It's a great environment. We have great facilities too, so I think it's going to be a lot of fun. So feel free to contact me. We're hiring at all levels. And with that, I will thank you again for inviting me to take part in this seminar. Brad and Mark, my postdoc advisors, funding, And with that, I will end and am open to questions. Thank you. I will have a drink.
Thanks very much, Yoav. That was a fantastic talk. I really enjoyed it. And it seems like people did too, because we got some really great questions that I think will prompt some into interesting discussions. We'll try and get through as many as we can. So one question here from Eamon Fitzgerald is, who says, beautiful data. Do you know if these neurons in the insular cortex are multimodal, i.e., do you see individual neurons that respond to both hunger and thirst?
I would say that we've done some experiments on this. I guess Sam maybe knows a little bit about the history of doing this. We've done some crude versions of these experiments, and the answer seems to be yes and no. So some of them seem to be specific, and some of them seem to be non-specific. We've been developing the system to do these experiments more carefully, with the mice being basically transitioning between consumption of solid food or water and basically changing in real time whatever they want to do. So then we can sort of follow the activity and follow the same neurons. And so those are actually ongoing experiments we're doing. And I think really the dream is deconstruction and the service of reconstruction. So the dream would be to know the so-called, tuning curves of these neurons. So to know whether the neurons that we see, for example, that increase their activity during satiety are these neurons that are responsive to gastric stretch and or nutrient absorption and or insulin levels. There are different things you can imagine that would modulate the activity. So we would love to be able to sort of parse out these things, and then that way understand sort of the global, because hunger and thirst, these are really, from the sensory perspective, they're very coarse. They're sort of like visual scenes in the visual world, but they don't tell us anything about, orientations of lines and colors in an analogy to that. So this is definitely this is basically part of the plan. So if you're interested, please join us.
Great. Yeah. So another question we have here from Vasil is, do you know if food with higher nutritional value would evoke a stronger decrease in the signal you were showing towards satiety in the population activity of insular cortex?
Yeah, that's so the short answer is I don't know, because we haven't done that experiment, but I definitely think it's a worthwhile experiment. And there are even simpler versions that we could have done, and I shamefully admit that we did not do early on, which is, for example, just manipulating the reward size, so a small drop of water, a big drop of water. I mean, I think nutritional value is a more elegant way to do it, but we basically don't know. But I mean, based on the results that I showed you, I think what you're getting at, and I agree, is that it should, and I hope it does. But if it doesn't, that'll also be interesting, and we'll be curious to understand. Basically, we don't know exactly what's being predicted. And I think this gets at that question, which I think is a fascinating one.
Mm-hmm. Yeah. So another kind of question, this one about potential roles in anorexia and eating disorders generally. So Juicy here says, exciting presentation. So what's your prediction about the functional role of insular cortex and, hang on, sorry, just jumped. And the power of food-related visual cues during hunger in a human pathological condition such as anorexia nervosa?
Yeah, so I mean, it's a great question. Again, I'm sorry to sound like a broken record. It's a great question, and actually a lot of people have been and are working on this. So especially, obviously, in the human neuroscience domain, there's a lot of work on this, and it seems that there are modified responses to food cues in insular cortex. I think one of the challenges is about, again, this multimodal nature that I tried to illustrate a little bit with this graph, of different lines going in different directions. Because, for example, there's beautiful work by Nadim Gogola's group and other groups showing that insular cortex is involved in emotional regulation, for example, encoding aversiveness or an aversive sensation. So, for example, in anorectic patients, there's a lot of work that suggests that a food cue would actually be aversive, at least under some circumstances. So the question is, how do you actually interpret the activity, right? So you can basically you would predict different things, and I think that's what the literature shows. It shows different things, and it's a big challenge to do it. And again, a lot of very smart people are working on this. Also, I should say, Mark Anderman, my ex-postdoc advisor, has some work on this, and he's trying to tease this apart in a mouse model, which again has its challenges, but has the advantages of maybe being able to tease apart activity patterns in a different way. So I'm sorry if it's an unsatisfying answer, but I think it's really open and exciting field for investigation.
Mm-hmm. Mm-hmm. Another question here from Thomas Pomberger. He says, "Great talk, Yoav. I'm curious, which part of the insular cortex did you record from, or is the whole insula involved?" No,
that's a very good point. So there's this idea in the field that there's this gradient from posterior to anterior in the insular cortex, and there's some evidence, of course, topography, but it's debatable, of going from sensory to associative. So we intentionally were doing all our recordings in the sensory part of the insular cortex that encompasses, in sort of a partially overlapping area, both the primary gustatory cortex and the primary visceral cortex. They're partially overlapping, and most of our imaging was exactly in the part where they overlap, where you're basically going to get a lot of everything. And the more anterior part of insular cortex is, eventually you get to lateral orbitofrontal, and there's a lot of work on that. So you can see anterior insular cortex in the literature, which is a lot of the times very close to being lateral orbitofrontal. So that's a much more sort of higher order associative structure. And there are a lot of fascinating theories. You can check out this review from 2015 from Lisa Feldman-Barrett and Cal Simmons about the sort of interaction between anterior and posterior. So that was a very long answer. The short answer is we were imaging in the sensory part, in the mid-posterior parts of insular cortex. Mm-hmm. But we're very curious to look at the loop between the anterior and mid parts.
Mm-hmm. So another question here from Malaziu is related to the data that you just mentioned very briefly, where you look at how the kind of time course of the changes in state encoding, let's say, in insula. So they say, very nice. What do you think is happening between the decline of insular cortex activity and task disengagement? If insular cortex activity reflects physiological state, how come the animal still eats or drinks in response to the cue, even after the insular cortex neuronal activity changes, towards the quenched state?
Yeah, the short answer is we have no idea. So all I can give now is wild speculations. It was truly, not in like surprisingly, like you read in the paper. Truly, that was one of the more surprising results for us, was the fact that it actually changed a little bit before behavior. And when I say a little bit, in neuronal timescales, it's huge. It's like 5 minutes, which is, if you think about the timescales of spikes, right? It's crazy. So we have no idea. It seems to be related, such that when the insular cortex activity started transitioning to a satiety-related pattern, actually predicted on a mouse-per-mouse basis when it's gonna stop doing the task. It just happened a few minutes later, but it actually was predictive. So the sooner the mouse has stopped doing the task, the sooner the insular cortex activity would also change. So there's some relationship between them. And why, you can say that there's overconsumption, there's sort of intentional, I don't know if that's the proper word with mice, but intentional overconsumption because of the severity of the deficiency. And I don't know, there are a lot of other speculations. I guess I shouldn't speculate too much, but I think it's a fun question to ask. And what we want to actually do now, and we're gearing up to do, is do a patterned activation of these patterns. So we can now, we have newer tools these days. We as a community have tools to activate specific patterns of activity in cortex. So the idea would be to identify the satiety-related pattern and just activate it. And maybe you can even tune the activation like halfway, all the way, right? And activate that pattern and see what it actually does to behavior with delays, et cetera. So I think there's a lot to do here. And we just want to, rather than speculate too much, we want to just do the experiments and see how it relates to behavior.
Yeah. Nice. So yeah, we're running a bit over, so we have quite a few more interesting questions, but I'll just ask one more here from Carlos, who says, fantastic talk. Predictions are very useful when comparing with the actual outcome. So do you have an idea of how that could work? And do you think that's done with an insula too?
Yeah, no, that's, I love this question. So basically, that's, in a way, that's the problem. I mean, there's a lot of appeal for working with states like hunger and thirst, but I think that's the problem with them, that they don't give you this type of resolution. Because what's like a prediction error of thirst, right? I mean, you could eliminate, you could you could do things like eliminating the cue, so having a cue, eliminating the reward. But what we actually want to do, as I said, is have proper stimulus control. And I think once you have good stimulus control, you can really do sort of the fine experiments of creating predictions, so you can track the activity before predictions are made, as they're being created, and then violate these predictions in different ways. So you can omit the outcome, you can switch. If you have two outcomes, say calories, no calories, you can switch them, or different macronutrients, you can switch them, right? So I think that's the type of experiments we want to do to actually understand what's being predicted and how are prediction error signals computed with prediction signals. What's the source of these? What's the circuit mechanisms for computing them? So this is if you read my research statement when I was applying for jobs, that's exactly the experiment I was proposing, or a series of experiments to understand exactly how all these things happen, the different circuit components of them. So this is, I think, this is definitely, I think, at the top of the list.
Okay, we'll sneak in one more question, and then that's it. So this question is about the role of orbitofrontal cortex. So the question is, the orbitofrontal cortex has been shown to have a role in creating anticipatory autonomic responses to external cues. So how do you see these two cortical structures working together?
Yeah. So again, a speculation, of course, I wish I could do all the experiments I'm imagining, but the way we think about it, or one of our, I would say, working models, is that what insular cortex, so insular cortex does different things in a way with this physiological state and coding. So, for example, it may use these predictions to regulate bodily functions, as in the Pavlov example I gave, but it may also use the current information to give the context for cue outcome associations, right? Because physiological states are definitely a strong part of the context for sets of cue outcome associations. And there's nice work also showing that the hippocampus, for example, as part of involved in encoding context, is important for knowing what to do. So you can train rats to do one thing when they're thirsty and another thing when they're hungry, and the hippocampus is important for this memory of context. In this idea, there's this idea by Schoenbaum, Yelniv, and others about orbitofrontal cortex encoding task states. And our speculation is that insular cortex contributes one component of that state, task state, which is the physiological state of the body in that state. I'd use the word state too much, but yeah, that's the idea. So it's part of the context. That would be sort of the bottom line of my answer.
Great. Okay. Well, thanks everyone for the great questions.
Thank you.
Thanks, Yoav, for joining us. I really appreciated it, and it was a really fantastic talk. Next week, we'll be joined by Pierre Leopold, who will tell us about development in Drosophila and how inter-organ communication and specifically neural signals control the body size of Drosophila during development. So looking forward to that. I hope many of you will be able to join us then. If you've signed up for the more extended Q&A session, you will have received an email from me, and we'll meet there in about 5 minutes or so. So yeah, hope to see many of you then, and see you next time. Thanks again, Yoav.
Thanks, Sam. Thanks, everyone. Where am I going, Sam?
I sent you a Zoom link by email.
Did you? Oh, okay.
Yeah.
Thanks, everyone.